All right, I'll give another minute for everybody to get connected here. Abigail, can you hear me okay? Hi, Jason. Hi, Ellie. Hi, Michaela. Hi, Roland. Hi, Tori. All right, can somebody say something? I want to make sure that I can hear you guys if you go to say something.
Hello.
Oh, thank you, Abigail. I'm like, nobody's volunteering. Okay. Liz, can you hear me okay? I can't see you.
Yeah, if you remember from before, my computer's broken. So
right.
I need a new one. I'm trying to wait out till graduation.
That's all right. Okay. You're all good then. All right. Thank you for meeting early. I'm miserable right now. This head cold is pretty bad, and I still have another meeting at 9 o'clock now still. So I'm going to try to get through this one, and then I got to get through another one at 9, but I do have a hard stop at 9. So I want to make sure to get started here so that I can make sure to hop onto that meeting. So units 8, 9, and 10, catalysis, kinetics, intro to metabolism. Do let me know if I start getting glitchy. My computer seemed to be slow when I was doing stuff. Do I seem okay or? Okay. All right. Very good. All right. So I'm going to share my screen and we'll sort of do what we've done before. I'll kind of go through. The only thing that I hadn't posted yet is the homework for the homework key for unit 10. That again is due tonight. So I will show you a homework key for that. And then I actually will probably post that one. It's a student from previous years. I forgot to make a copy of mine before I left today. So here is kind of an overview of content. Let me zoom in here a little bit. Sort of split up into terminology and numbers, concepts, function, and applications, which is, I think, what I had it split up into for our last learning unit, and kind of orienting you again, anything that's listed with a number dot something, that's indicating the learning unit, and then the homework question that's associated with that, so you could sort of find something. So I'll sort of go through things. I'll try to go through and do enzyme catalysis first. So I might jump around here just to kind of do enzyme catalysis first, and then we'll go back and do kinetics and then metabolism. And then we'll highlight and I'll go through and see if there's anything either in the homework or in the notes that I didn't cover sort of on the study guide here. So again, thinking about enzymes, we learned about a reaction coordinate, be able to draw and discuss the relevant parts of a reaction coordinate and how they equate to how enzymes catalyze reactions. For the enzymes that we learned about, be able to identify the enzyme names, the substrates that they participate with, linkages that they cleave, things like that. And a lot of these that I'll flip to the homework in a minute so we can see these homework questions. That's the purpose of the homework, is sort of to take some of this stuff and have more of a meta-analysis and sort of bring stuff together. Certain amino acids have catalytic roles. So being able to identify what those amino acids are capable of doing, whether they're general acid, general base, covalent catalyst, electrostatic catalyst. You'll sort of see that there's going to be maybe a group of 7 to 8 amino acids that can participate in those roles. And again, those are the ones that have functional groups. Understanding the different modes of catalysis, and then serine protease names and substrates. So that was the only one where we really had to associate trypsin, for example, cleaves after lysine and arginine residues. Elastase is small nonpolar, and chymotrypsin is those bulky aromatic side chains. So that's sort of those pieces. I'll go over to the homework in a second, and we'll kind of see some of these pieces there, but I'll pause for a second and see if anybody has any specific questions before we look at the homework. Okay. I'm going to just kind of stick with Unit 8 here. So conceptually, this kind of concept of thinking about the energetics of breaking and making bonds. This is a super big picture thing. Anytime you break a bond, you need to put in energy. So conversely, that must mean when you make a bond, you get energy. So if you remember it that way, if you think about breaking a stick, right? If I'm going to break a stick, it takes energy. So I can't have things go downhill both ways. So if you're going to make a bond, that must mean you get energy. Conceptually, from an energy coordinate, right? If we bring and overlap electrons together to make a bond, that becomes something lower in energy. We talked about sort of the energetic problems with a lock and key model. That's a very nice conceptual model to think about why we have enzyme specificity, but it really fails from a thermodynamic standpoint, and the induced fit model is more relevant there because of entropy, right? Pre-organization is entropically unfavorable. This is sort of a big one here, but the concept of being able to think about how we detail enzyme active sites. And so there was a series of 3 questions on that. And again, dealing with those 4 enzymes that we studied, RNase A, carbonic anhydrase, lysozyme, and serine proteases. There is one that you haven't seen yet. So you will see detail me a step of one of those. And I will tell you, it's not carbonic anhydrase. We really don't have a nice enzyme active site for that. So it's either going to be RNase A, lysozyme, or serine protease. You're going to see one of those steps. I will show you the details of the enzyme active site, and then you have to describe in words using that Mad Libs what's going on. Okay. Strongly encourage you to understand how that process sort of happens in terms of writing that detailing, because you are going to see an enzyme active site you haven't seen before, and I want you to do the same thing. And then there really wasn't anything application-wise, specifically in enzyme catalysis. So I'm going to go over to our homework, and we'll kind of look at some pieces there. And then I'll flip through the notes and see if there's anything that I'm noting there that I really didn't make a comment on. My computer's
Dr. Prene.
Yeah.
When we get to that part, can you do an example of just like a Mad Libs situation when we get to those parts?
Yep.
Thank you.
So obviously, and this might be slightly different in terms of the numbers from your homework, because I think when I made my keys, I had a few more questions, and then I just never rewrote my keys. So just keep in mind that some of these questions might not be here. Again, these 3 are sort of related. Breaking bonds takes energy, making bonds makes energy. Enzymes catalyze reactions by lowering the transition state. We kind of see that in words here, but see that with a reaction coordinate. Be able to draw a reaction coordinate. Think about delta G double dagger. Think about delta G, and how enzymes then catalyze reactions. Hopefully this is sort of a pretty straightforward way of just kind of putting together all the information for the linkages, the monomers, the enzymes, and sort of the location and function for these 4 enzymes that we studied. So again, these were already homework keys that were up there. So hopefully, and again, if you have questions on any of these in terms of something that is not clear from the key that I have here, let me know that. For the most part, there's a short list of amino acids that are able to participate in catalytic roles, right? It has to have a functional group that's there. So we need to have things that are either positively or negatively charged, or have a heteroatom that can be nucleophilic. So that's what all of those guys are. Vitamins, right? We had a fundamentals quiz on that. So sort of knowing specifically as they relate to, you're not going to see the entirety of your fundamentals quiz on there, but any B vitamins in particular that have relevance to things that we studied, I would make sure that you know those. Describing the 4 modes of catalysis, right? General acid, general base, covalent catalysts, and electrostatic catalysts. So again, here's that Mad Libs. If you see, and so Ellie, if this sort of helps, if you see an enzyme active site that you haven't seen before, look for where the arrows are. They'll either be an amino acid that you should be able to recognize what it is because you see the side chain there, or it'll just say, whatever it is, or it'll show that specific atom. Look for where arrows start, specifically if they start at amino acids, and then apply your Mad Libs. Tell me who that residue is. What is it doing? Is it adding or removing a proton? Is it forming a covalent bond? Is it serving to stabilize charge? And then tell me the chemistry that it's performing. Is it forming a tetrahedral intermediate? Is it making a good leaving group? You know what? Just try to look at the picture that's there, look at the arrows for what's happening, and tell me the chemistry that it's performing. Okay. Understanding that, and you won't have an entirety of an enzyme active site in terms of all the steps, but when we sort of role played these, we saw that there was generally 2 acts, right? The first act is we do chemistry in the substrate. The second act is we have water coming in and basically regenerating the enzyme active site. So chemistry on the substrate, using all the reagents, which often is water and the enzymes that we've learned, and then returning the enzyme to its active state. So I'm not going to take the time and sort of go through all of these. There's videos that are online. You know, the notes very clearly sort of run through each of these individual steps. Again, I would highly discourage you from just trying to blanket memorize these, because then you're not learning the skills that are going to help you apply this to, you know, a new system. And you don't have to get the words exactly right. You don't even have to have the syntax exactly right. You just sort of have to have those pieces in place that describe what you see happening in an enzyme active site. And like we've indicated before, your enzyme active site is always going to be what it looks like before and after an arrow. Okay, so that's going to be the step that you'll sort of detail. So you'll look at the enzyme active site before the arrow, and that's what you're going to be describing is happening to generate what you see after the arrow. Okay, so there it is for RNase A. Carbonic anhydrase, I do want you to know some details about carbonic anhydrase, but I promise you that is not the one you will see for detailing the enzyme active site. And then we had lysozyme, and then we had serine proteases. And I don't know why, I guess I don't have, am I missing a page here? You have more after this, right? Don't you have serine proteases? I don't know why my homework key doesn't have that. Is that missing on the homework key? Do you know? All right, so I will have to find that. Let me actually go over here and I'm going to try to find this here. Let's see. Enzyme catalysis. Just going to open an old student homework here and I'll see if I can find that here. I'll make sure to sort of look through. Okay, so here it is for serine proteases. Again, nothing that's should be unusual here. I'll make sure that I either post this. I guess it's just that's the last piece, right? Just the last one is the serine proteases that's missing. But I will make sure to post. I'll check over this question 15, and if this student has everything right, I will make sure to post that. Any questions on the homework from unit 8 there on catalysis? Let me just go back here to slides and see if there's anything I want to go through. I'll flip through these kind of slowly too, or if there was a slide that you're like, hey, yeah, I don't remember that part really well. Can you go through that again? So there's reaction coordinate. We talked about that. Kind of an overview of, we're talking about polymer hydrolysis for the enzymes that we've studied. Looking at the uncatalyzed reactions for RNA hydrolysis, carbohydrate hydrolysis, peptide hydrolysis. There again is the lock and key model. Induced fit helps remedy that fact that we have unfavorable entropy if we are freezing out an enzyme active site. We talked about modes of catalysis. Again, there's your Mad Libs, sort of how to describe that. And then we sort of went through each specific enzyme. Liz, I'm really appreciative of you of like sitting down and working with me to sort of like act out those things. I think that did a really nice job of trying to illustrate what I wanted to do with all of you guys, sort of putting on the name tags, but can sometimes create more confusion than I want. So again, there was a homework question that said, where do we find these or why do you have them? So I do want you to know, like, yeah, where do you have RNA A in your body and why do you have it? Or these other enzymes. So again, I'm just kind of flipping through here, making sure that there's nothing that I haven't touched on. Specifically because we didn't talk about carbonic anhydrase, I do want you to understand this concept of metal binding water and that inductive electron withdrawal when we have coordination of water to metal ions is what actually makes metals more acidic. So you probably learned this in general chemistry. If you don't remember, that's okay. But when you have a highly charged metal in water, it'll actually make that solution acidic. We need metals sometimes to do a variety of different purposes in enzyme active sites. So just remembering what some of those things are. Sort of the neat one is that we can do a lot with metals that we can't do with amino acid side chains. So we can do redox chemistry. We can also do a lot of the same things that amino acid side chains can do. So we can kind of stabilize or shield negative charge buildup with those positive metal ions. We can bind and orient substrates. And importantly, this piece is key because we can affect sort of the pH, or we can affect the amount of hydroxide we have here without affecting pH. All right. Lysozyme, again, some details with lysozyme have to do with sort of some unique substrate conformational changes. So we've talked about sort of what that is with that half chair. Again, we bring in energetics to sort of think about how that sort of plays into things. Again, as I'm just going through and giving a real rough summary, if there's something that you want me to say a little bit more about, just speak up and I can kind of explain a little bit more. So that was lysozyme. Serine proteases. Again, it's a class of enzymes, right? Because we have substrate specificity that flanks that cis-aryl bond. And so knowing these names are important and the substrate specificity that they correspond with. There was the Mad Libs for that. We talked about having the oxyanion hole, which is a way to stabilize negative charge development as we go to a sp3 hybridized tetrahedral intermediate. And then we've already learned about proenzymes before. We talked about proinsulin. So zymogens are proenzymes of proteolytic enzymes. And we talked about the importance of making sure that you don't make protein digesting enzymes in a protein containing organ like your pancreas in an active form. That would be very problematic. So not only do we have sort of a domino effect of being able to activate these, but we have sort of this mechanism that if anything were to get out into your bloodstream, we have this essentially suicide inhibitor that can kind of help remedy things. All right. So that's a quick overview then of Unit 8 on catalysis. Any questions on that before I move on to kinetics?
Is there a mechanism on this exam?
Nope. Essentially, your mechanism is embedded in detailing an enzyme active site, but not drawing a mechanism. All right, so moving on to kinetics, right? And I'll highlight some of these things when we get to your homework. You do need to know the Michaelis-Menten equation. So not only the enzyme reaction equation, which is enzyme plus substrate, goes to enzyme substrate complex, then chemistry happens to make product plus free enzyme, but how that maps into the Michaelis-Menten equation. So the terms that we see in the Michaelis-Menten equation, substrate concentration V naught, Km and Vmax, so be able to define what those are and discuss their limits. We talked about regulation via activity generally in this learning unit. Very briefly, we talked about availability. You can upregulate the synthesis or regulate the degradation of enzymes as a way to control their availability, but a much more effective way is to control whether an enzyme is on or off. And we do that through a process called covalent modification, adding or removing phosphates with kinases or phosphatases, and then structural allosterism, which is the binding of small molecules that toggle between R state and T state. So big picture, we've seen this several times throughout the course. I want you to be able to connect phospho versus dephospho systems with need versus fed states. Okay. Nothing it looks here that I have in this concept section. Sort of from a function standpoint, big things, thinking about kinetic plots. How do we determine reaction velocity? How do we determine that initial velocity? And then how do we translate that into a Lineweaver-Burke plot? So we'll look at those details in the homework key in just a minute. Understanding how both competitive and uncompetitive inhibition work. And then sort of the big enzyme that we sort of studied in that learning unit was to understand how glycogen phosphorylase worked. So that was the first time we really looked at an enzyme active site and tried to understand how enzyme structure affords function. So kind of from an application standpoint, we talked very generally about how many drugs are designed after enzyme transition states, because we know an enzyme is going to bind the transition state most tightly. So if we want to design an inhibitor, we want to make it look like the transition state, and we can measure the affinity of that inhibitor for the enzyme by determining its Ki value. Okay, so I'm going to go over to the homework right now. And let's see here. I'm going to get rid of this guy. And I'll get rid of this guy. And go over to homework here. My computer is really slow. Hopefully it's not too laggy on your end. All right. So, and my numbers might be a little bit different than the homework that you have, just because this is my key from a few years ago and I didn't rewrite stuff, but for the most part, it should be the same problems, sort of understanding and being able to write this general equation for enzyme kinetics, and then writing and understanding the parts of the Michaelis-Menten equation. Okay. Understand the limits that we have for each of these, right? And understand sort of what happens to this equation and the effect on V naught as we think about large or small values of Km, large or small values of substrate concentration. And specifically, you can do the math to say that when the substrate concentration is equal to the Km value, V naught, simplifies to 1/2 Vmax, which is the mathematical definition that we know for Km. Any questions on that initial part? Okay. In the laboratory, measure things that we can measure, right? So we can measure oftentimes the change in concentration of either a substrate or a product. And so you can either show that as concentration or you can show it as absorbance, knowing that Beer's law can allow us to relate in a linear way absorption and concentration. We would predict higher substrate concentration here. And again, if we're looking at the appearance here of a product, let's say, higher substrate concentration would make that product appear faster versus lower substrate concentration. But the key is that the slope of this straight line, particularly if it's only linear in this early region, is going to give us the rate of the reaction, or V naught. This is what I like to call the intuitive plot, the plot that makes sense as we increase the concentration of substrate. Because remember, this really, there's 3 plots that are on here, but when we just do a single plot with absorbance as a function of time, we're looking at one substrate concentration. And so that's going to create one of these points here. As we increase substrate concentration, our reaction is going to get faster. And theoretically, we are going to approach Vmax. But we can't solve in a simple way this equation to get what Vmax is. So by plotting as a double reciprocal, we can get things as a straight line. And then from the y-intercept, we first determine what Vmax is. And since the slope is a ratio of Km over Vmax, once we know Vmax from the y-intercept, we can use the slope to determine Km. Questions? All right, moving on, thinking about inhibition, okay? I want to make sure that you know how this equation is going to be different when you consider a competitive inhibitor which bifurcates the pathway here, versus an uncompetitive inhibitor, which is going to bifurcate the pathway here. Okay. Most of the rest of what we see is going to be the same in terms of defining what Ki is, thinking about what alpha is, things like that. Okay. I want you to be able to think about, for both competitive and uncompetitive inhibition, how Km and Vmax are affected, right? In all cases, an inhibitor is going to make Vmax be smaller, but Km appears to be larger with a competitive inhibitor, but we have an unchanged Vmax. We just need to eventually outcompete the inhibitor. Be able to think about how these plots look, both sort of our standard intuitive plot, as well as a Lineweaver-Burke plot under competitive inhibition. And then again, that alpha is really a scaling factor. So alpha is equal to 1 plus the ratio of the concentration of inhibitor over Ki. And so think about what these limits are. If we want alpha to be powerful, we want to have a powerful inhibitor. 2 ways that can happen. We can have high concentration of inhibitor, and or we can have a small Ki value. Both of those are going to make this term be significant such that alpha will be much greater than 1. So the range of alpha is from 1 when we have no inhibition, theoretically, to infinity where we have the highest amount of inhibition. If you want to figure out what your alpha is, you can take a ratio of your slopes, since Km and Vmax essentially are not changing. This is the apparent Km. So the Km, why it's larger, is it's scaled up by that alpha factor. Okay, so that's competitive inhibition. Noting how uncompetitive inhibition is different. Again, we're going to have inhibitor bind to now the enzyme substrate complex. So this is a little bit harder to think about conceptually because it does not affect the ability of the enzyme to bind substrate. If anything, it makes the substrate bind enzyme more tightly. In that case, Km decreases. Okay, Vmax is also going to decrease because we've handicapped our enzyme. Okay, thinking about how these look, I didn't save room here, so I'm going to come onto this next page, right? We have a similar looking plot, right, for our sort of intuitive plot. And it's hard to distinguish competitive and uncompetitive inhibition looking at this plot because we really don't know where these lines end up. So looking at a Lineweaver-Burke plot is going to be the best way to distinguish competitive versus uncompetitive inhibition, because you'll see parallel lines for uncompetitive inhibition. You'll see lines that converge on the same y-intercept for competitive inhibition. I may have misspoke there. Uncompetitive inhibition is having parallel lines. So that kind of gets us through regular Michaelis-Menten kinetics, competitive inhibition, and uncompetitive inhibition. Questions on any of that? All right. Then we talked about regulation of enzymes, right? And so I already sort of defined these terms when we looked at the summary, thinking about regulating availability, which is sort of a long-term strategy, versus activity, which is a short-term strategy. 2 ways that we can control activity that we've learned about are allosteric, structural allosterism, or structural or allosteric regulation, which is binding a small molecule that toggles between R state and T state, or phosphorylation, which can often modulate that same R state, T state conformation. Again, this hopefully is a review thinking about phospho versus diphospho systems. So phospho systems, we have high kinase activity. We're decorating things with phosphates. We are in a glucose or energy need state. So here are some of the signals that you know at this point for those. A drop in blood sugar levels, increase in adrenaline, increase in glucagon. A diphospho system means we've got elevated phosphatase activity. So we're going to be snipping phosphates off. Again, that's fed state. Markers for that are going to be elevated levels of glucose and then high levels of insulin. So all of these things sort of track together to make sure that we've got this logic check for what should be happening. And then the last piece of this learning unit here that we had on enzyme kinetics was understanding the structural regulation for the enzyme phosphorylase. So phosphorylase is the enzyme that breaks down glycogen. So here again is the equation for it. You're basically taking glycogen, you're going to break that glycosidic bond by phosphorylasis. So you're using phosphate as a nucleophile. You're getting glycogen that's shortened by one residue. And because we're using phosphate and not water as a nucleophile, you don't get free glucose, you get glucose 1-phosphate. Again, this would be happening under a phosphose system. We shouldn't be breaking down glycogen if we're in a fed state. And we would predict then that this enzyme would be R state. So considering the fact that we've got R state, T state, phospho, D-phospho, there's 4 possible sort of combinations of states that we have. And we looked at the next picture here in just a minute, but understood that it really sort of funnels itself into 2 states. We're going to be R state and phosphorylated, or T state and dephosphorylated. And we can look at the structure of that enzyme to understand why. And that's this picture, right? So there's a couple of pictures that you're likely to see on your exam, and you need to be able to explain, if you don't see pieces on here, which one of these is T state, which one's R state. How do you know, right? In this case, R state is determined by the position of the N terminus. If it's external, it's R state. If it's buried, it's T state. And at its core, it has to do with whether or not that serine 14 is phosphorylated, right? Phosphorylation of that serine 14, not only does it mean we don't want to bury a negative charge, but it also means that we have the ability to lock it into this external position by electrostatic interaction with an arginine residue that's here. So I think that's all the information that's kind of indicated here by sort of circling some pieces. Let me see if there's anything else to sort of add here. I don't think so. I think that's sort of it.
Quick question.
Yeah. Hi, Cassie.
Is R state phosphorylation?
So R state, good question. So R state does not necessarily mean phosphorylated. R state generally means that the enzyme is going to be on because you are in a conformation that's conducive to bind substrate. Okay. Now, for this enzyme, R state, meaning that it's on, so it's going to be binding glycogen and breaking it down, we need to do a logic check and say, ooh, if I'm breaking down glycogen, that must mean I need glucose. If I need glucose, I must be in a need state. Need state means phospho. I must be decorating things with phosphates using kinases. Right? So we need to be able to do that logic check to connect those pieces together. Okay.
Yes,
All right.
thank you.
Yep. I'm going to flip through the slides real quick here. They're back over here. Just make sure there's not anything that was in slides here that I need to discuss again. I know this learning unit in particular, I did a number of videos last year that hopefully can sort of help, particularly with understanding Michaelis-Menten kinetics, the different aspects of enzyme inhibition, whether it's competitive or uncompetitive, and then particularly the control of glycogen phosphorylase. So I'm just going to flip through here. I'm going to have to get something. I'm going to have to get a cough drop in a second here. I'm going to see if there's anything that I haven't mentioned that's here. There are a number of equations that are on here that are not equations you need to know. And I've usually tried to make sure when we went through this in class that I indicated those. Again, this is not, you don't need to know anything about K-cat or turnover numbers. Suicide inhibitors are ones you can sort of think about it as sort of just an inhibitor that binds irreversibly. Okay, they often are competitive inhibitors, but they don't necessarily have to be. It's just something that you never can fix it because it sticks on permanently. I think we've discussed all of these pieces. So again, just a couple of examples of competitive inhibition. So alcohol dehydrogenase activity can use both methanol and ethanol as substrates. And so we sort of talked about this as an example of competitive inhibition and how you could use an IV of ethanol to treat methanol poisoning. And to Cassie's point, we sort of, address this slide, and make sure you just sort of know this slide is specific to glycogen phosphorylase. So in this case, it's saying that a phosphorylated enzyme is on, but whether or not a phosphorylated enzyme is going to be active or inhibited, we have to know what that enzyme does, because knowing what that enzyme does will help make that logic connection with whether or not it's going to be active in a phosphorylated or a dephosphorylated state. This was also sort of mapping on some stuff that we already know about glucose transporters, about whether or not muscle cells versus liver cells are able to sort of facilitate, glucose export. So sort of our first introduction to a substrate cycle. Again, here's that picture looking at T state versus R state. And don't worry about drug design. This was just meant to be sort of an application piece. I do want you to understand that we design drugs to mimic transition states. But other than that, you don't need to worry about the clinical trial piece. I do want you to know that the best drugs are going to be ones that have small Ki values, because otherwise you have to take a lot of it in order to have it have its inhibitory sort of function. We really didn't talk too much about cytochromes, but just remember what they are. Again, cytochromes P450 are a class of enzymes that basically decorate foreign substances with hydroxyl groups to make them more polar. Okay, we're going to see them later on as well. So this was sort of just like a first read through, a first kind of examination of these. And we talked about it in the context of a Tylenol overdose, that we sort of have a maximum amount. We didn't talk about what the excretion pathway was, but you sort of have a maximum amount of Tylenol that can be excreted by this pathway. And then once you've sort of swamped out that pathway, then you start tagging in glutathione. And that can be problematic because that can lead to liver failure. And that was it for this learning unit. Questions? All right, so moving on real quick to the stuff that's hopefully freshest in your brain. And I'm going to get through this in about 15 minutes here, so we have enough time for questions, and then I can hop on my other meeting. Let me grab cough drop real quick there. All right, we'll just try some.
Dr. Prente,
Yeah.
is the insulin case study, like, are all the case studies going to be on the exam, any of them?
Not specifically. So let's see, what did we have for these? We learned about serine proteases. That was your enzyme catalysis case study, right? So it was how we inhibit those with PMSF. Your case study for the kinetics was an Excel workbook. So I'm not going to be asking you to think about anything like graphing data, but I do want you to be able to look at a graph and understand by the shape of things, whether it's like competitive or uncompetitive. And then we did study alcohol dehydrogenase. So yes, but no. I'm not going to be asking you to, like I said, do the graphing piece like you had on that kinetics case study. Does that help?
Yeah, so it's basically more of like what we already learned in the lectures. Like it
Yep.
ties in with the case studies, but focus on the lectures.
Yes, absolutely. All right. So learning unit 10 is that intro to metabolism. There's like a lot of big picture stuff. And then we have details of understanding pKa, which we just finished today. So those notes are up on Blackboard. So we talked about like the values of different fuels, linkages, monomers, and so forth, and that oxidation continuum. So again, we'll look at the homework questions for that in just a second. Four main processes that we use in fuel digestion, right? So we were very carbohydrate focused. So we talked about glycolysis, pyruvate dehydrogenase chemistry, citric acid cycle, and then electron transport and oxidative phosphorylation. We sort of took, and I would say, you know, put a little star by this. That's sort of a big picture that ties stuff together. We started out biochemistry by learning that main fuel equation. And now we understand a lot of how, you know, those simple products or those simple reactants of fuel and oxygen are ultimately converted to carbon dioxide, water, and energy. We talked about substrate cycles, and today we talked about near and far equilibrium sort of systems and reactions. So I'll make sure to kind of highlight that again, since that was something we just did very quickly today. Your homework is going to, you know, do a good job of sort of highlighting the relevant details that you need to know for these 4 steps: glycolysis, pyruvate dehydrogenase, citric acid cycle, electron transport, oxidative phosphorylation. And we also talked about lactate dehydrogenase and alcohol dehydrogenase as fermentative pathways. We only had like single slides on fat metabolism and protein metabolism. So again, your homework does a really good job of highlighting the pieces that you need to know from that. And then the 2 branch point metabolites that we focused on were pyruvate and acetyl-CoA. And then big picture thing application was understanding how that adenylate cyclase pathway in its synthesis of cyclic AMP ultimately regulates the activity of pKa. Okay, so I'm going to go over, and I haven't posted yet. So I have a student example of homework. And so I will make sure I will post this later tonight after I sort of confirm that everybody sort of uploaded their homework, or at least the majority of you have. And then I will make sure to get mine uploaded tomorrow as well. So this should be pretty straightforward. Again, a way to sort of have a meta analysis of the fuels that we've discussed, the 4 processes in carbohydrate metabolism. So again, asking where they occur, how much ATP is produced, and whether or not any of these small molecules or cofactors are made. There's again that main fuel equation that's expanded on. So I'll show you that slide in a minute. Talking about the digestion of fuels, right? Think about some of the names of the enzymes that we talked about that are responsible for that. How these are delivered to the bloodstream, specifically lipids. We talked about there needing to be lipid particles that they transfer, or they or why can I not think? Proteins and lipids together. So, and I know, and I will highlight, I've sort of made sure, I don't know if I've adjusted it yet in Blackboard, but homework from learning unit 9 and 10 are going to be worth more points because there is just more effort that you had to put into them. So those will get reconciled at some point when I have a chance to fix it on Blackboard. It's not easy to do. You need to do it sort of one at a time. It's kind of a pain in the butt. But, you know, this is just kind of a nice way to summarize the slides that we had in sort of a consistent way. So you know the nuts and bolts and the meat of things that you need to know in terms of what's the net reaction, how many steps is it, where does it occur, anaerobic or aerobic, if there are stages, what are the main things that happen in those different stages. So I'm not necessarily, and I will post this, I'm not necessarily going to go through each of these ones individually. If there are things that are confusing to you or you want me to sort of go over, I can. Just going to see if there was anything here that jumps out that may be a little bit confusing. Or if anybody's worked on the homework yet and you have questions on stuff that you weren't certain what I was asking. So again, this is the stuff we just talked about today. And since it was probably towards the end of class and your mind was probably about full, just kind of summarizing the concepts with near and far equilibrium reactions. So if something is near equilibrium, the delta G value is close to 0. Far equilibrium are large and negative. Okay. Why this is we've got an R state enzyme. It's kind of like a leaky valve. So it's easy to just kind of put things in the forward or reverse direction in there. And it's always on. And so it's not good to have that as a control point. A far equilibrium reaction, the enzyme actually is in T state. So because it's been put into a T state and it's kind of off, everything upstream of it sort of builds up. And so we're building up thermodynamic pressure, which is why it's large and negative. And so because we have the ability to turn the faucet off at that point, that's why it's a control, it's a control point, because we have the ability to turn that enzyme on and off. So here are some of the things you will see, something that's sort of related to be able to discuss a sort of cascade for me, right? So connect your understanding of pKa to its role in glycogen metabolism. And I don't think there's a separate part here, but again, glycogen metabolism really only has to do with need state. So when you are in a need state, you have a stimulatory signal on a protein receptor. That might be glucagon, it might be adrenaline, any of these stimulatory signals. Ultimately, we activate adenylate cyclase. That's going to increase the concentration of cyclic AMP. A little bit more detail probably in here is we are going to bind that cyclic AMP to the regulatory subunit. Right up here, we bind that cyclic AMP to the regulatory subunit. So the regulatory subunit dissociates, and we are going to have those free catalytic subunits that are able to do their chemistry. So by activating pKa, it's going to phosphorylate, and this student doesn't have that intermediate piece. pKa actually phosphorylates phosphorylase kinase. Phosphorylase kinase is what phosphorylates glycogen phosphorylase, and that allows us to break down glycogen. So I want you to be able to think about putting in sort of a little flow diagram of what happens to connect a need state, low blood sugar, to an outcome, breakdown of glycogen, that fixes that. And that's sort of the big picture of where we're headed, is to understand how signals ultimately translate into biochemical processes that help remedy whatever that sort of signal was. Questions on anything from the homework there? I'm going to go over for a few minutes here to the notes, and we'll just see if there's anything you want me to address there. So here are the notes from our metabolism learning unit. And again, these were ones that either you already had digitally because you're a digital note taker, or I gave you sort of them. So hopefully it's something, got it. I will go. Who's iPad 153? Is that you, Cassie? Okay. Go over branch points again. Yes. So we spent a good bit of time on this slide. Try to recreate this slide for yourself, right? If you were to go to the notes and look at a blank version, could you fill in any place that there's an arrow? Could you discuss what's going on? Remember, this is a story that you're trying to describe for how we ultimately take fuel and generate energy and then waste products. So star this slide, right? How do you take this key equation and sort of map on the details for what we know for how this works? Let's see, I'm just going to slowly flip through here, see if there's anything I want to add. But Cassie, I'm going to get to your point. And even though we have all of this information on this slide about glycolysis, right, your homework is going to do a good job of having you answer all of the relevant questions for what I need you to know about glycolysis. So those two pieces can sort of talk to one another. We talked about all of this. So Cassie, the key with a branch point metabolite, let me actually come up here just for a second. These two are the two branch point metabolites we talked about, pyruvate and then acetyl-CoA. So why we call them branch point metabolites is because there's lots of things that can feed in or out of this molecule in a biochemical system. So they represent, in sort of a metabolic pathway, places where we can branch off and do something else with that molecule. So that's why we call them branch point metabolites. And the two that we've learned about right now are the 3-carbon molecule pyruvate and then the 2-carbon molecule acetyl-CoA. And why those two are important to think about is because pyruvate can be made into acetyl-CoA, but not the other way around. So once we've committed something metabolically to acetyl-CoA, we can't go backwards. So hopefully that helps, Cassie. Here's that energy continuum piece, right? Be able to use this then to explain why fats are worth 9 calories per gram, but carbohydrates are only 4. They're more oxidized, so they're not worth as much because they're already further along on this oxidation continuum. And a lot of this are things that we saw someplace else, but since they have a home in this learning unit, we've sort of like pulled all of these pieces together. So a lot of the stuff that's in this learning unit is new, but a lot of it may be things we learned someplace else, but it's like, hey, do you remember when we learned about this? It's got a home here, right? So that's the case with NAD plus and FAD, with ATP production. Cassie, hopefully that explains and answers your questions about branch point metabolites. Big picture, I want you to think about fates of different molecules under need state versus fed state, right? Talked about substrate cycles. We talked about near and far equilibrium. And then this was the last thing that we sort of talked about. We didn't learn these details, so don't worry about them. But ultimately, when we have a stimulatory external signal, like glucagon, like epinephrine, we're going to activate adenylate cyclase. Adenylate cyclase is the enzyme responsible for making a cyclic AMP. Once we make cyclic AMP, we have the ability to activate pKa. Activating pKa as a master phosphorylator. This is the cascade that I want you to know. pKa phosphorylates, phosphorylase kinase, phosphorylates serine 14 of phosphorylase, and phosphorylase breaks down glycogen. Okay, breaking down glycogen is going to give you glucose that you can put into your bloodstream. And that ultimately is going to remedy if our stimulatory external signal to start was glucagon. Okay, and hopefully this is fresh in your brain. We looked at sort of the structures of the catalytic subunit, and I know that there's videos that are online that sort of run through this pretty well. So structures of the catalytic subunit, the regulatory subunit, and then how they play into either a need state or fed state sort of system. Questions? And unfortunately, I think my voice is about shot. So I don't know how much I'd be able to talk more anyways. I have to get myself through this next meeting as well. Questions. I will be around tomorrow. Emily has her senior thesis presentation at 1. So those of you who are biochem majors, I really encourage you, if you're able to, at 1 o'clock, come support both Gabby Ialachi and Emily Parker as they sort of have their senior thesis presentations. They're the 2 seniors that are graduating a little bit early, so they're giving their senior thesis presentations this semester. Emily is going to be talking actually about malic dehydrogenase. She was a student in my lab. Gabby was also a student in my lab, but she's actually going to be talking about a summer research project that she did as an internship. But those are going to be tomorrow from 1 to 2. So point being, I will also be around from 2 to 5. My biochem students, or not biochem, my gen chem students have their lab final. And so I probably could sort of quietly try to have a conversation if you have some questions about something. And then in the morning, I don't know when I'm going to be in, but I know Emily needs to go through and practice her talk some more. So I don't know how available I am in the morning. So maybe the afternoon might be the best time to sort of try to find me. And certainly you can send me messages via Teams, and if it's something that I can sort of address quickly that way. All right. All right, guys, I have to hop on to this next meeting momentarily. I'm going to try to make myself another cup of tea before I do. But I will post this as soon as it's finished here. There's also one from last year where maybe I don't sound as awful. Probably has the same information. I'll stick around if anybody has anything, but we'll see you guys tomorrow, and if not tomorrow, Wednesday. Oh, I should say too, I'll send this out in a message. You're welcome to come and start early at 8:30, just like we did before. Or if you need to stay after, you can do that as well. We'll probably need to find a different space for you. But so you can either start at 8:30 or stay until 10:30. Sorry, 10:30 is what I meant to say. All right, we'll see you guys.